Okay, then we'll start. Welcome to the this week's class on fMRI data. So we'll talk a little bit about processing and quality control, and hopefully people can hear me okay on Zoom. I guess I'll open up my chat window in case people have questions. Or Pete, would you mind monitoring chat? You do? Okay, then I'll defer for that. Okay, what I'd like to do is, starting with this, is I don't know, has everyone looked at fMRI data in their time here or previously? Has anyone not? Perfect. Okay, we have one person who has it. So I'm going to start off just looking at a little bit of data. And so this is from one of the boot camp classes that we teach. So generally, whether you're doing a lot of fMRI analysis or any other kind of project, it's always good to look at your data first and get a sense of what you're going to be dealing with. So one of the first things that we look at when we're acquiring our data is typically when we're processing fMRI data, we have an accompanying set of data that provides structural image. So this is what's called a T1 weighted anatomical. And as you can see here, it has good anatomical information. You can see the different parts of the brain, the different tissue types. The white matter here is light and the gray matter all around. If we hop up to the ventricles, we'll see the ventricles are dark. And this kind of data helps us recognize all the different parts of the brain, and that's fine. But this is not really the functional side of things because we don't see it doing anything over time. This is a static three-dimensional volume. And when we're looking at this, we talk about 3 dimensions, we'll look at it in this kind of rendering as 3 different planes. So the axial plane, the sagittal plane, and the coronal plane. And actually, I'm just going to turn one of these planes off just so I save a little disk space or screen space. And there's text in the terminal. We don't need that at the moment. When we're going to be looking at fMRI data, I'm going to load up in what's called an underlay here, an EPI data set. So it's acquired with MRI in a slightly different way. And those of you who've used the AFNI GUI, which I'm sure is everybody or will be by the end of your summer, we can see that this is a 3D plus time, and there's 152 volumes here. When we look at this data, we can see that it has pretty different properties. And actually, let me be a little bit smarter about this. I'll keep the other one open. I can open up another GUI where they're locked together so we can keep them at the same time. All right. So in this case, as I click around in the brain here, and the crosshairs are locked, but immediately we can see just spatially, this looks a lot fuzzier. It doesn't look nearly kind of as nice. If you saw this image walking down the street, you might be able to recognize them. This one, it's probably a little bit harder. One thing to notice is that the different tissue contrasts are a little different here. So here in white matter, it's the kind of brightest part here. In this image, it's the darkest. We're not really concerned so much with that, but the fact that you can see the kind of just image quality, right? This is your modern day iPhone, and this is probably the iPhone that I still use, or an older model or something like that. But yeah, just a lot grainier. Another thing is that this is nice geometrically. This is not really distorted from how it's acquired. It has a lot more fidelity to the actual person in the scanner. This one, maybe you can see a little bit that the front of the brain shouldn't really do this anatomically, or shouldn't have this kind of wedge chopped out of it. That is a geometric distortion from acquiring the data. So overall, you can also see that we've chopped off part of the brain here as well, whereas I see basically the whole head, even outside the skull. We don't see the skull here, but we can see that the top part of the brain, the person, much like myself, has had a close haircut recently. Unlike me, they've also had their cerebellum chopped off. So we've traded off a lot of things when we acquire this data. Why are we even bothering acquiring this? Because everything we've said is kind of negative. Well, the reason is I can open up a graph window, And I see that in addition to this spatial information, that's not really what I care so much about here. It's the fact that I have associated with every single location in the brain, a time series. And if I zoom in on one, we can see this. So this means wherever I click around in the brain, what we're doing as we're acquiring this upper volume is we're acquiring it very quickly. That's why it looks larger voxel, so less clear. It's a little more distorted, but we did that because we want to acquire a whole brain image every 2 seconds. And what we can do is if I hit the mouse arrow over here, or just the V key, you can see that this is what the brain looks like as a movie. And essentially what we're doing with fMRI is acquiring a movie of the brain, where it's kind of snapshotted every 2 seconds or so. And the details of how that's done and the variability, there's a lot of different options that go into it. One thing that you may have noticed already as we're watching the movie, right now you can see it changing a little bit. It doesn't change very much overall. It looks mostly pretty constant with how we look at it. But then at some point, something happens a little more majorly here. At this point in the movie, it's moving, the subject is kind of nodding in the scanner. So if you allow your subjects to move and don't kind of fix them very well or things like that, this can happen. And this is something we have to deal a little bit with in processing. What we want to do when we look at fMRI data is we're interested in the time series here and all their properties. There are maybe 2 major camps of fMRI acquisition: task-based fMRI and resting state fMRI. In resting state fMRI, you put someone in the scanner, you tell them to just be relaxed, and you scan them for 5, 10, however many minutes. And what you're going to look at is how similar the time series are in different parts of the brain, suggesting that they're working together in some way. With task-based data, which is what this is, there's no way to know it just by looking at it. I know it from prior experience. What you do is you put someone in the scanner and then you have them do something that you're controlling from outside. A classic example from the early days is you shine a light, and while you do that, you tell the subject to tap their finger, their fingers or their hands while the light is on, and when it's off, they stop. And when you do that, you're looking for maybe motor cortex lighting up, something like that, to see which parts of the brain are involved with that particular task. Nowadays, tasks tend to be a lot more complicated. They can involve lots of different things via very short durations. So then you're in the realm of doing linear algebra to kind of pick out information from your time series. As I click around here, so if I just look here, and let me just make it a little, here, let's look at a 3x3 grid. What do you see when you look at these time series? And you just think of what are we looking at here? Without knowing too much, how would you describe these things? Does it look well planned? Does it look random? Does it look large? Does it look changing, constant in time? You can think to yourself, it's a little bit hard with people on Zoom and here in person, but these are the kind of things, when you're looking at data, these are properties you might be considering. To me, this looks fairly random. It's kind of flat across time. It's not really going up or down or getting larger or smaller. Okay, that's fine. Now, if you think about where we are in the brain, even though it's a little bit hard to tell, if you know your anatomy, we're kind of sitting in white matter. It's fine. Something to notice, if you can kind of see at the start of every one of these 9 time series, there actually is a feature that's here where it's kind of up high and then drops down. And here there's a bit of it's a little bit hard to see where the line is. But this is something that happens with fMRI physics, with the way we're acquiring the data. What we really care about is this part where it's fairly consistent and flat, kind of bubbling up and down in the same range. This part in the beginning is due to the fact that the magnets have to kind of reach saturation as they're starting. So this is something we'll see often. Sometimes the scanners delete the first few time points, but these first few time points are actually different. All right, if I click around a little more, I'm going to click over here to some part of kind of around gray matter. You probably notice some different feature in the time series now. There's a giant spike. Importantly, this spike is not always in one direction. Sometimes it's up, sometimes it's down, sometimes it's really big, sometimes it's smaller. And I guess I should have mentioned here that this axis is looking across time here. If I kind of click in time to where the spike is, and I move in time around, there's a little red ball here telling me what time point we're at. You can see what is causing that spike. It's the fact that the person's head is nodding a little bit. And if you think about it, what's happening is we're measuring something in space, right? Someone's head is here, and you imagine like you're a little monitor sitting at this voxel right where the crosshair is, at kind of this location in space. And then the subject's head moves. So now you're monitoring the kind of the brain activity there, and then the subject moves, that part of the brain moves as well. So the thing you're monitoring just moved away from where you are in space in the scanner, and now you're monitoring something maybe very different. And usually where we see spikes is where you've been monitoring like gray matter, subject moves, now it's kind of white matter that's there, and maybe the brain moves back or whatnot. This particular nodding motion looks fairly much like kind of back and forth, like you're measuring something, subject moved, and they kind of move back to where they were, so you're back on course. And that's why the spike looks fairly clean. It's kind of bubbling along, subject moved, and you're kind of back where you were. Subject motion is typically much more complicated. This is an extremely good subject to only have something like this happen. Especially if you're scanning kids or older subjects, you really have to be concerned about how much motion there's going to be. All of this part that we're measuring here, all of this is the bold activity, the blood oxygenation level dependence. And so these are the things that we care about that's kind of related to neuronal signal. This spike is physiological, but it's not really information we're going to be caring about in our analysis because it's something different than what the brain is directly doing. I should say if there are any questions, just ask them. I'm happy to take questions throughout on anything. All right, and if I keep moving around the brain, let me go to this part of the brain. We still see spikes. Are there any other features that you see? Anything else that you would notice about some of these time series that's a bit different than what we've been looking at so far? Yeah, there's something different. Mostly what we've been looking at so far looks like this, or maybe like these. These two here are decidedly different. They have something else happening there. There's kind of a tooth or kind of a box car, right? They look like train cars coming by. Still noisy, but there's something else like a definitive pattern. And indeed, this is something that's actually a hallmark of the task that we've given the subject to do in this case. The subject has been given an audio-visual task that's on and off for, I think, 20-second chunks. It's part of a kind of mixed audio and visual. If you know, which I don't really know my parts of the brain very well, I'm a lowly physicist masquerading here, but this part of the brain is the visual cortex. And so it would make sense that this part of the brain is involved with the visual task. Now, I didn't tell everyone it's a visual task, but now you know it is. And that's why I knew when I click back here that some parts of the brain might look something like that. So I can also turn on here and show you a little bit about what the actual paradigm looked like, just to prove a little more about this. So let's see here. I actually have stored in this file an idealized time course based on the on-off pattern that the subject did something. And in this case, it is an audio-visual task. What would their idealized, hypothetical, purely hypothetical, idealized response look like? It would look like this red curve. And if I pump up the signal a little bit, you can see that indeed this boxcar pattern, not only is it kind of matching the on-offness, it's synced with it pretty well. Okay, so this is what we're going to do with our analysis now. Basically, what are we trying to do with processing with task fMRI? We gave the subject something to do. We want to find the parts of the brain that look like they're doing it. Not every part of the brain looks like it's involved here. If I go to white matter, right, that's just a complete mess. This part of the brain doesn't look like it's very involved in that task I gave. If I jump back, this part of the brain really looks like it is. And even if I go, it's not only just all gray matter. If I go to this gray matter, it doesn't really look like it's involved here. That's fine. Not all of the brain is doing everything all the time. So anyways, essentially what we're trying to do with our analysis and our processing is we want to basically get at this question: what parts of the brain look a lot like the time series we gave? And we want to avoid features that are kind of non-physiological and noisy, like this spike, right? If we just do simple time series correlation between one of these time series and this red curve, this spike is a major feature that would throw off our measurement. It would kind of disrupt the correlation that we want, or the regression that we want. But this is really going to be the goal of our processing: get cleaner estimates of what part of the brain look like they're responding to the tasks that we're providing. So that's just setting up like what our data is, what we're trying to do. Any questions on that? Now, I will note that if you're analyzing data this summer, if you're analyzing task fMRI data, it's probably not going to look quite like this. Modern tasks are not so on for 20 seconds, off for 20 seconds. Modern designs tend to be more event related. So you're doing something quickly, then quickly, and randomly out there. You may also be clicking buttons and responding in different ways. So now you're going to be analyzing not just the brain functionality on and off, but analyzing responses. How fast did you reply and things like that. This can get much more complicated quickly, and it's a little bit harder to eyeball. Is your data making sense at all? Whatever you're doing for your data processing, it is nice to start off to kind of get to know your data a little bit in the beginning and see, do things make sense? Are there weird features here? There are weird features here, the spike, we can understand it and we'll try to account for it in our processing. Actually, we've seen two spikes so far. The one in the beginning that's always up, and we'll deal with that. And then this one that occurs during the time series, and we'll try to deal with that. Okay, so yeah, okay, zooming out a little bit here. So just to talk about setting up the analysis. So when we want to do our analysis here, okay, sorry, it's a little chopped off on this screen somehow, but there's not that much missing. What we want to do now is we want to think about our processing as this set of steps of cleaning up the data, getting rid of bad things we don't want in there, and kind of digging out or accentuating and preparing the things we do want in there for our analysis. And so you can think about that as an algorithm or flow chart about different things we want to get rid of. So for example, I think, yeah, despiking appears here. This one, we've seen a couple occasions where spikes occur, and those are going to be things that we think are obviously not part of our time series. We've seen it bubbling along, and then things that are obviously outside that range. Slice timing correction deals with the fact that in reality, I said that every 2 seconds we take a picture of the brain. It's not just poof, picture of the brain, and 2 seconds later, poof, another whole picture of the brain, but we're actually acquiring bits of it over time for 2 seconds. So a slice here, then here, then here, then here, and for that entire 2 seconds, we're kind of filling up the whole volume, and then we start doing that process again. That process doesn't just go slice by slice through the brain nicely. It usually interleaves or more complicated patterns just because of the physics. We want to deal with the fact that the brain is moving. It's nodding. So if we imagine measuring our time series, our little monitors are at constant points and the brain moves away and maybe moves back. What we want to do is we want to try to capture the fact that the brain moved and when we estimate our motion, we want to estimate how much motion that was and maybe try to move it back where it should have been, so that time series that was in that same part of the brain that moved away, we can grab it and put it back where it should have been. We're going to do some various alignments. I'll show an example of why we do alignment. And then we're going to go to what's called standard space, which is part of spatial normalization. So looking at this data here, this person's brain is kind of this own person's quirky brain, right? The motor cortex is here, the number of sulci and gyri and things like that. If you're scanning someone else, their brain has different sulcal and gyral features, and so does everyone else's brain here. If we're going to do a research study, we want to be able to click on one part of the brain and say, this is the motor cortex for everyone. But since everyone's brain is a slightly different shape and has different features, what we're going to do is we're going to take this and mush it around to a standard template brain. In that way, we'll be able to click on one spot and everyone's motor cortex should be there. If I just took this volume and everyone else's brain scan here, because some brains are a little bit taller, shorter, narrower, different shapes, it wouldn't work to kind of match up features. So that's essentially what we want to do with that spatial normalization. Even between these two brains, if I go here and I overlay my EPI brain now, and I try to render this, you can see that these brain scans that we took consecutively while the subject was in the scanner, they line up pretty well, because the subjects, when they're in the scanner, they're not really allowed to get out and go back in, but their heads can move around a little bit. They relax between scans, and it's fairly fine, but kind of the overall edges are pretty good, but we can actually do better. So we can make sure that the subject's EPI, fMRI data is actually more well aligned to their own anatomical. Why do we do that? Because we want to kind of leverage the different benefits or the different main features of each kind of data set. This data set doesn't have good spatial features. So if we want to try to warp this brain to a standard template, if we try to warp this, it's fairly ugly and not very detailed. This data set, on the other hand, has nice details. So this is one we can align with a standard template brain well. If we can align this one to this one, and then this one to a template, we can chain together those transformations, and our fMRI data ends up in final space. So even if the details are maybe not clear if you haven't been kind of working in this quite yet, this is pretty typical for most data processing. You're going to clean up various features and maybe transform it in some way. In this case, there are a couple different spatial transformations that it undergoes. And then finally, one of the main parts about this data is dealing with the actual time series themselves, which is what basically all of this does. So this pipeline is referring to resting state data, but it's essentially most of these features are the same. We're going to be performing a general linear model to clean up the data, remove features like subject motion or baseline changes that can happen in different things, and try to get at a measurement of what we want. So all of these features here happen in a regression step. As with all of these things, these are the kind of general ideas, and the details matter a lot for how you do it. You can do these things well, or you can do these things poorly. In the same way, you can write an essay that kind of contains the same major features of something. It can be well written, or it can be poorly written. I won't go too much into maybe all the details here, but basically, we estimate all these things individually, and you could either apply them individually or as a concatenated single one. We apply it as a concatenated single one. These are the kinds of things that are maybe not biological questions, but people who look at the kind of math and physics and the algorithms think are important to improve the quality of the data here. Okay, and there's a lot of different ways that you can try to get rid of extra things in your data that you think aren't related to brain activity. So subject motion is only one of these. There are a lot of other things in fMRI that affect your data, and affect it in the sense of what you want is blood oxygenation response specifically related to neuronal firing. But you have to remember that you have blood oxygenation response related to things like breathing, heart rate. Those are very hard to turn off in the scanner, right? Or you don't want to volunteer for that study. There's all sorts of other things that you want to try to like pull out of your data. So you're focused on that idealized or as close to what you think is the idealized response to neuronal firing. So these are acronyms that you may or may not be familiar with, but that's what they're trying to do. Okay. Now, at the end of the day, these are a lot of steps of things to do. And I don't know, are people here programmers? Do you enjoy programming, scripting? Okay. You can write your own programming script to go do all these things. There's a software package like AFNI, which is what we're discussing here, that you can do all of that individually, which is great. Or you can use tools that are called pipeline generating tools. So that's what this one is here, that maybe looks fairly complicated, but it has nice features of essentially what you're doing is setting up that whole processing, and it looks like a lot of options, but it's actually kind of specifying not too many things when you think about all the things that have to happen or trying to program it yourself. Here, these are just the input data sets. These are the two data sets that we were just looking at, an EPI data, which is the fMRI data, and the subject anatomical, which was the nice clean one. These are the things you specify, like your major processing blocks. First, I want to despike it. Then I want to take into account the fact that it's not a whole volume instantaneously, et cetera, et cetera. And so it's nice to break your processing up into the kind of major processing things, and then later specify the details, because that way it's just a little cleaner to conceptualize the bigger picture and then focus in on each. And then this is where you focus in on the details of each. And the way this is organized is, if you notice the start of most of these option names here, all match, most of them match the name of one of the blocks here. So if one of your blocks is volreg for volume registration, these are two options that go with it to specify what you want to do specifically. For regression, there are more options here because that's something that requires a little more control. And maybe by not specifying options for these other ones, you're happy enough with defaults. So that's the way you would read this. Another thing that's a programmatic point here, these slashes that appear at the end here, when you analyze something either from a file or from a terminal, you basically take everything that's in one line as part of the command, and then you hit enter, and that's the end of it. If you want it to continue on to other lines so that it's a little more spaced out rather than just wrapping around awkwardly in your document, what you can do is you can put these continuation of line characters. So this is just one giant command that we keep saying, keep reading to the next line, but it's visually, it's not too bad because they're all lined up vertically. Okay, at the end of the day, what does that get you? That doesn't just start processing your data. What that is going to do here is it actually creates a script for you that you can read that describes everything about how your data is going to be processed. So this is something where you say, dear AFNI, I would like to do these steps with these options, please do it. If it just processed from here, you wouldn't know all the details yourself, the exact programs, the exact numbers put in. And if somebody asked like your boss or a colleague or something like that, you wouldn't be able to answer them. So the fact that this creates an exact script that says, this is in detail exactly what I'm doing is great. Another thing to note here, and you can, even if it's a little hard to read, the coloration will point out something important. All the red text here are comments. So not only is this just all the commands that are being run, they're guided. The script is automatically made to have comments in here. So this AFNI-Proc program specifically was written by Rick Reynolds in the group, who is, right, he's written a command that gives you something that generates a whole commented script, which is a very nice thing. So I don't know, anyone who's ever written their own scripts to process data, one of the things you always swear you're going to do is comment your script nicely for other people in the lab, for yourself, so generations can admire it. And of course, we never do. This problem has been solved for us, so we can benefit from that. And then something else we'll take a look. Well, okay, let's pause there and look at a little bit of processing then. So I'm going to close this. And just to show you for the data that we're going to talk about here, what does the processing script look like? It's the same kind of thing. We can load in more than one EPI data set. So you can have the subject, take 5 minutes and do something, relax a little bit, do another 5 minutes, another 5 minutes, and we can process it all together. In this case, this shows maybe a more realistic example of, as you get used to processing data, you want to control a lot more features about the processing, and you can do it. So we kind of put out vanilla examples for processing, like if you're starting with task-based data, here you go. If you're starting with REST, here you go. And then you can take and tweak everything you want. That's fine. But anyways, so this is a task-based data set with 2 types of tasks, an audio and a visual stimulus. And you can not only tell the program to kind of estimate what parts of the brain are responding to the visual stimulus more or audio stimulus, but you can make general linear models contrasting them. And most fMRI analyses nowadays involve multiple types of stimuli while the subject is in the scanner. And you're not so much interested in just what is the visual response, what is the audio response, because those tend to be pretty well known. What you're interested is, what is the difference between audio and visual, or the difference between congruent and incongruent Stroop tasks, or seeing numbers versus letters, or colors versus black and white shapes. And it's those differences, those contrasts that are of interest, so you can estimate those while you're processing. Just to mention here, again, the script that gets created, so it is fully commented. You can see it a little bit better here. And so anyways, if you have any questions on your processing, you can see what's happening in literally every step. So if you want to know what happens in the volreg block and why, you can search for the start of the volreg block in the title here, and it tells you all the different things it's doing and the options and why. So actually, if you do want to learn about AFNI programs and see a lot of them, this is actually a convenient way. I often refer back to the kind of automatic generated script for reference. Okay, so what happens? Oops. When we process our data, what do we get out of it? Well, like I said, we want to find these statistical maps, and we can look at those quickly. If I look at what's here in what's called the results directory, there's actually a huge number of files that are here. There are 194 files and things here. Now, some of the files have like pairs of data and header files, so it's actually less than this. But there's a lot of things here to sift through. This is because, in general, there's a lot of different philosophies for when you're processing data. What do you want to do? What do you want your result to be? Well, I just want my answer, so I go on with my life and write my paper, right? That's the idealized thing. The more you're in analysis, you realize you kind of want to make sure that all those steps that we asked the program to do got done right. And so you want to be able to check that if you're trying to align 2 things, they actually got aligned, right? Just because the program didn't crash doesn't mean it did that right. Or the fact that you want to make sure that the subject didn't move too much, or things like that, or there aren't other mathematical problems here. So we tend to keep a lot of intermediate data. So once you've processed it, you can check anything that you have a question about. open here. So at the end of the day, you can look at things and you can kind of jump to, for example, the statistical map here. Bring it to the forefront. So I'm just going to jump to the results for a second to see what we're getting at. All right, so this actually is the results of all our processing and what we're trying to kind of look at when we have fMRI data. If you remember, we used to have time series everywhere in the brain. And then we said, what do we want to do? We want to see, well, which of those parts of the brain are involved with different tasks. And at the end of the day, we get out a statistic map and an effect estimate map of where that is for everywhere in the brain. So this data set looks a little different. It's been aligned to a standard template brain. So I'm underlaying the subject anatomical. And this is the result of our general linear modeling. It looks kind of messy. One thing you'll notice is there's results everywhere, not just in parts that overlap where the brain is. We have signal that we've estimated everywhere, and we can perform general linear modeling to basically ask the question, which parts of the brain look like they're involved with my visual stimulus? So I can see here that this is the part of the statistics, the coefficient and the statistic related to the visual part of my fMRI data set. Now, without knowing too much about the data here, you can see that the bright red and the dark red and the dark blue are where things are the largest, either positive or negative. As you look at this data set, what parts from our modeling look like they have the largest coloration here? Outside the brain. So that means that the air around my brain is actually pretty good at this visual stimulus, right? So even if I turn my head, I'm still seeing you because I have this halo of activity, right? Well, that's what it might look like here, which is a funny kind of thing. And what we're looking at here are the effect estimates of our modeling. This is another large topic, but let's suffice it to say that what we're looking at, what the coloration means here, and this is -6 and this is +6, this is kind of bold percent signal change that we've estimated. Okay, so it looks like our largest effect out here is like ±6% or maybe more, but it's outside the brain. That's a little funny. We know that the visual cortex is kind of involved. If I go here, part of the visual cortex is also red, so that's good. So we've done a scientific measurement and we've seen the largest numbers are outside the brain. That should probably worry us a little bit, because we would probably expect to see where those up and down boxcars were. We saw those aligning well with our stimulus inside the brain, and everywhere else it looked very garbagey. So why is this happening and what can we do about it? Well, whenever you take a scientific measurement, you don't just want to get the answer. Like if you take a poll of people, do you like cheese? 50% of respondents said yes. Okay. Obviously, this is not done in the great state of Wisconsin, which would be much higher. But is that the end of your measurement? Is that what you report? There's another piece of information that's really, really important about that. Are you going to go buy stock in cheese based on just that? What else do you need to know about your measurement there? Or any scientific measurement. You get a number and you say, hi, this is what I estimated. Plus or minus what? Is it plus or minus 2%, plus or minus 0.5%, plus or minus 50%? That error estimate really matters. So whenever we do a processing or statistical modeling, we want to get not only our effect estimate, which we can do here, but the plus or minus on it, the error bars. And that's what this other piece of information here is. So I can actually choose to threshold by it. So I'm going to lift the threshold up on what's the t-statistic information, which is essentially related to that plus or minus. So as the statistic gets higher and higher, the uncertainty of what I'm looking at is getting lower and lower, the standard error. So I'm going to put it up here to a little over 3. So now what we're seeing is two pieces of information from our measurement. We're seeing where the effect estimate is big or small based on the color, and we're seeing where it has small standard error relative to that based on now I've thresholded the data. So now what do we notice about where things are? All of that stuff that had big ±6 that looked like, big effect estimates outside the brain, that also had giant error bars, essentially. The statistics there were very small. Conversely, you could say that the uncertainty there, the standard error was very, very big. So the reliability of those numbers is very poor. So we kind of combine these pieces of information where our effect estimates are large, where our uncertainty or reliability is low, where the reliability is low, let's say. Okay, now what is the correct value? As I lower it here, right, it's a sliding slippery slope here. Maybe I push things up here to a very high statistic. I still see some things. I'm probably missing some things that are kind of actual here, right? fMRI data is actually pretty noisy. So we don't really know exactly what the correct statistic to threshold is. Do we want a threshold at whether you want to think about plus or minus 10% or plus or minus 2% or 2.1%? There are some kind of maybe guidelines or approximate numbers that people like to use, but those are always arbitrary. So something that we do when we're checking out data is we say, not only show me where the statistics are high, so I see those, but what we can do is we can turn this threshold, instead of seeing nothing out here, we can turn on what we call transparent thresholding. And this is a nice way to make our results, as we look at them, less dependent on the actual threshold that we're using. So we can tell if things are slowly dipping down below threshold or not. So when we look at our data and kind of go through for sanity checks, this is something we would do. So transparent thresholding means, instead of just hiding absolutely everything that's below threshold, even if it's 0.00001 below threshold, this means that we let its opacity just fade as it gets further and further from threshold. So it's like if you put your hand below water, you can still see it, but as your hand goes further and further below water, right, it starts to fade away. It's that exact same thing, just with statistics instead of your hand. And then we kind of, to put extra emphasis around things that are above threshold, we can do that to highlight them. Another thing to look at here is we're looking at percent signal changes of blood oxygenation with fMRI. The color tells us like the amount of bold percent signal change we're seeing. What should we expect to see in an fMRI experiment? Should we expect like 20% bold percent signal change, 50%? How much should we expect change? There's baseline fluctuations. The brain is always moving. When we give someone a task, what do we expect percent-wise? I'll give you a hint, 20%. Going to 50% is the wrong direction. And if you remember, even here, all that stuff outside the brain was at 6%, right? Everything here is much smaller. Actually, what should we set our color bar range to? Maybe the most we might expect is something like 2 or 3%. That's actually a really strong response for a very strong signal. This goes along with the phrase, fMRI data is very noisy. We put someone in the scanner and we give them a very big, obvious task to do of 20 seconds of strong visual stimulus, 20 seconds off. The most that we're seeing in response to that is like a 3%, maybe 4% change. And this is really, kind of in the heart of the visual cortex. There's an audio component. That's why this is red. So when you're processing your data, you have to know what your expected signal to noise is, or how much bang for your buck you're getting while the subject's in the scanner, and kind of set your expectations based on it. So if we were looking for something at plus or minus 50%, we wouldn't have it here. So this is now, we can kind of look at our statistical results for a sanity check. And what did we do? We had an audio-visual task. So what would we like to see? We'd like to see audio and visual parts of the brain with strong responses and strong statistics. So depending whether you know kind of brain biology or not, or fMRI, that's what this map kind of looks to me, because when I'm in the visual cortex here, I see this red, which is now set to be 3%. And I know that that's about the most I can expect. Yeah, that looks reasonable. And there is an audio component. It's actually a mixed audio visual. So even during the visual, yes, so seeing something in the auditories also makes sense. Now, I'm seeing lots of stuff elsewhere. I'm still seeing stuff outside the brain. Should I be worried? Maybe. Am I worried here? Probably not so much. Even seeing stuff outside the brain, that's probably not too problematic, because we're looking at blood oxygenation response, and near draining veins, or even near like skull that's near the brain, sometimes you'll kind of see that signal appear in places you might not expect it, in kind of an expanded range, because we basically have slightly magnetized fluids going through a magnetic field. Kind of odd things can happen a little bit. And again, there still is just noise. So purely random things, even here, this is above the threshold out here still. Our data has been smoothed out there and some other things, but data is really, really noisy. So what do we do to kind of improve our studies with fMRI? We don't just scan one subject and say, these are the parts of the brain doing it. We scan 10 or 20 or 1,000 or more, and we pool all those results, and those will sharpen our statistics. Those little random bits that appear at the edge and all those things, those are random in every different subject, and those kind of fade away when we combine the data. So fMRI typically requires group statistics. But you want to know, are these noisy bits that I'm seeing here problematically noisy, or just kind of expected noisy? And that just takes experience and kind of looking at data to get used to the difference between those. So are there any questions on this? Now, to get to here, we've kind of jumped from like my raw data, we look at the time series, and we said that, you know, that flow chart of things. So there are lots of intermediate steps to look at to deal with kind of checking those intermediate steps. We could go through the files and look at things. But another thing that AFNI creates is an HTML report of lots of different features of the data. So everything kind of matters with your data from start to finish to get those statistics there. So we spent some time in the beginning looking at the time series, which is great, and you should always do when you're starting a study. Some of the things that we might look at here are just checking to make sure that we acquired, like, this is our EPI data, just a single volume, and the QC is written so that there are these montages of pictures of the data, so you can quickly see everything and check for kind of good or bad. What are things we'd want to look at here? One, that we got the whole brain that we wanted to. Like if we're doing a cerebellum study, and the cerebellum would be right here, kind of going down below, we would be sad. If we're doing even up here, we might be a little sad, because the field of view that we're looking at is a little probably too tight, because things at the edge are really affected and all that. So anyways, it depends what you want to study. If you're studying the visual cortex and auditory, you're probably okay with this. And you likewise want to make sure like that for your anatomical, you can check that they overlap well. And something that you want to check is, I said there's a step of alignment between the EPI and the anatomical. And when you want to check, did that work well? What you really care about are all the sulcal and gyral features matching up between the two. The way that we represent this here, because it's hard to look through one data set and see another one, is these are the edges of the anatomical image. So the edges of this nice clean data set shown over the EPI after alignment. So does it look like features match up here to you? Boundaries of ventricles, boundaries of sulcal and gyral patterns? To me, it looks like it's okay. Similarly, we align our anatomical to a template data set. So again, we'd look and see, does our anatomical, which is the underlay here, match up with the outlines of our template that we were trying to get to? Do the sulcal and gyral patterns match up? How do they look to you? They don't? Actually, I would say, yeah. I mean, you can tell that it's probably been aligned a little bit, but it could be better. If you look down here in this slice, it actually looks pretty decent. And like ventricle boundaries, you can tell that there's some overall general feature mapping, but you can see the lines here aren't great. And actually, when you're looking at the cortex up here, it really looks much more jarring. It looks like kind of maybe a kid scribbled with crayon a little bit. They don't line up tightly. Why is that? Actually, this is because this is our in-class teaching example that we run, that we want to run very quickly. So this was only a quick, not very detailed alignment to template. And so this is only a quick, not very detailed alignment. So this can and should and would be better in a full analysis. So full marks for skepticism. You don't have to compliment the data just because I'm up here saying this is our data. That's okay. Most people don't, in fact. This shows an image of where the data has pretty good SNR, basically, signal to noise ratio. Something you have to watch out for in EPI data is if you notice here, there's not very good signal here. And actually, we would have noticed that if we looked carefully up here, if you notice where the edges are of the anatomical, there's not EPI signal underneath here. This happens for a couple reasons. One, because our field of view is tight, and two, because you have these air pockets that are your sinuses here, and immediately above there, that part of the brain is terribly difficult to acquire good signal in, because having an air pocket there, just the way the magnetic physics works, you don't really get good signal here. So this is something that people really have to watch out for. If you want to study this part of the brain, this data is not very good for it because your signal is almost non-existent there. So these are the kind of things that even though the alignment is good, you see the brain overall, the details really matter a lot. Again, if you care mostly about visual and auditory, those parts are kind of colored in. There is some subtlety to the QC. If you want to dig in depth, you can click on some buttons that are here. So I clicked on this NV, and this is now an embedded browser thing called NIVU. Sure, they'd be happy to hear it called an embedded browser thing, but it's actually a pretty amazing tool. And you can search through and check different things. You can hit C, and you can kind of browse through here and move a clipping plane, so you can really investigate things in detail if you want. It's nice to have systematic images. It's also nice to be able to hop into it in detail. And then looking at the statistics, this is something that we looked at just a little bit. There's a lot of different aspects of the statistics. You can ask, is my model a good fit in lots of places overall? That's what the full F-stat tells you here. So one of your quality control items would be looking to see if you have basically colorful values here in your audio and visual cortices, which is good to see. The visual data set, if this looks familiar, it's because you were awake 10 minutes ago when we looked at this. Thank you very much for that. And this is the kind of thing. So that loading up the data, changing color bars, setting it, all those, that's been done for us automatically because we have a sense of what we want to look at, and it's much faster here. So we can look at the visual one here, and this was the auditory response. And actually here, you can see the visual minus auditory contrast. This is much noisier, but this shows you above threshold where there's a significant difference between them. In this case, where in blue, where audio was much bigger than visual. So this may just look noisy. That's okay. But as you get used to looking at these things, you build up more expectation of what should this look like. And when you see a subject that doesn't have it, it's easier to pick out. One of the most fun things that you can do while checking your data is to run what's called Instacore in AFNI. This is something that forms really what I would consider the heart of quality control in resting state data, but it's also useful in task-based data too. In this case, we want to look at basically spatial temporal patterns across the brain. So Instacore has been loaded, and what I do now is I hold down Control and Shift, and I click somewhere. And what it's showing me in my 3 different planes are what's correlated with that. Now, this is task-based data, so we're not strictly doing just correlation, but it's nice to see what time series look like other time series. If I'm in white matter here, this is good to see. White matter shouldn't be doing a lot of the functional processing. It should be where most of the cabling is. And it shouldn't be highly correlated with parts around the brain. It should be kind of locally bubbly, just signal kind of static in maybe an ideal world. So that's good to see. As I come down here to the visual cortex, I see a lot of the rest of the visual cortex lit up. That's also a good thing to see. If I come up here, let's see, yeah. If I look for correlation here, that I'm in white matter and I see something that's highly correlated than anti-correlated, like kind of a step ladder of correlation and anti-correlation. This is something that's a little bit weird and non-physiological. And this is something that actually we've come to realize through some QC projects exists in a surprising amount of data and is something you don't want to see. This is a scanner problem that's actually happening in the data. If we look at just what the standard deviation looks like of the data, you actually get essentially you have these high lines of variance going through your brain in the raw data set. And that's essentially what is causing these correlation, anti-correlation patterns. We don't actually know why, but it's something kind of related to a frequency in the room or a radio signal or something that's doing bad things to our data. So for a task-based data set like this might not be a problem. It fortunately kind of occurs in parts of the brain we're not probably studying, but for a resting state data set, this would probably torpedo it because it injects this very strong correlation, anti-correlation pattern. So anyways, the moral of the story is there's a lot of things to check in your data. You have to have some expectation of what should be there, and you want to check intermediate stages as well. Don't just look at your final results, because sometimes you'll see things and then you'll realize only much later, oh, this alignment step was bad. That's why I got activation here. I didn't discover something new about the brain. It was a processing mistake early on. And you also have to accept the fact that real data has real weird stuff, whether it's noise or minor artifacts, and you have to understand what's causing it as much as possible and whether it's going to affect the scientific questions that you're actually going to answer. Something weird happening in the front part of the brain, if you're only caring about the visual cortex, might be okay. But if you're doing a whole brain study, no. Something here, these correlation, anti-correlation patterns may be okay to see in a task-based study, but it'd be devastating in a resting state study. And basically the jobs of myself, Daniel, other people in the group, other collaborators, are to try to make it easier for you to check your data in intermediate steps like with this HTML, and also provide things that are like automated warnings, which I don't have time to talk about, that exist here, including these variance lines and things like this. So anyways, I think I've reached the end of my time. Thank you. Are there any questions? Yep.
About the ball signal in the red color map on the data set you show. What about the blue color?
In the statistic map or here? Oh, in the statistic, yeah. So yeah, so I focused on the red and hot color here. What about the blue part? That is a question here. To be honest, I'm not quite sure why this would be so strongly anti-correlated. I think the other part where I focused on it was in the contrast map here. When you're looking, so this contrast is visual minus audio. So, you know, it's literally where visual is much bigger than audio will be very positive, and where audio is much bigger than visual will be very negative. And so the blue here is where the audio response was much bigger than visual. And so, yeah, so it makes sense that we see that in the audio cortex. And here in part of the main part of the visual cortex is kind of hot color still, because the visual response is much bigger. Now, all of these other parts, certainly up here by the ventricles or in white matter, that's probably noise that won't survive group statistics. This part, let's say here, I'm not exactly sure why that is. Yes. Yes, if we look at it here, you can choose to threshold. So this is that. Well, this is sorry, this is just the pure visual one. Let's see, the one we're looking at was this. So here, you can threshold and choose to only look at positive or negative. In contrast images, most people look at both. Most of the times people are saying, where is there a difference between audio and visual? And then the hot color ones are where visual is a lot bigger, and the cold color ones is where audio is bigger. But certainly most people are just asking for where is there a difference, and then those two different sides of the color bar separate the directionality of the difference. So if I want to, I can turn off seeing the negative stuff. So now the color bar stretches from 0 here, so I'm not seeing anything that's negative, but most people actually look at it fully like this. Okay. Oh, is there a question in the chat? Just Pete. Okay, thanks. Okay.